How is everyone? Great, awesome. All right, well, for the next couple of lectures, what we're going to do is we're going to spend some time considering bacteria, bacterial structure, function, some specific enzymes that bacteria possess that makes using certain antibiotics very, very attractive. What I really want to do is emphasize the concept of selective toxicity to all of you. Selective toxicity being a concept whereby we can administer a small molecule or even a larger molecule to a patient and have that molecule go and target a specific enzyme or protein in a bacterial cell that is absent in the mammalian host. Thereby, we're just affecting or inhibiting some type of process in a bacterium without doing much damage or any damage at all to the host. So that's what we're really going to focus in on. So in my humble opinion, I think if we have a good review of certain features of bacteria, that provides us then with a scaffold to build upon all of this antibiotic knowledge that we're going to get into. So we can't just start talking about beta-lactams. We can't just start talking about fluoroquinolones if we don't understand what the role of those drugs are in inhibiting or killing the bacterium that we're trying to go after. So we're going to spend a little bit of time doing that today. First part, again, just probably a lot of review, and I'm okay with that if you're okay with that. And then the second part, or the second half of the slides, we'll get into the classes, which is kind of like the overarching goal of today. All right. So I like to talk, obviously, but I don't like to talk to myself, even though I do that often. So if I can engage in conversations with you guys and get you guys to talk back to me, that would be awesome. So here's a question. Which of the following cellular components are seen in bacterial cells but not in human cells? This should be like, boom, right? It's very straightforward. A cell wall, all right? The cell wall. So the cell wall is the fundamental difference between a mammalian cell and a bacterial cell, and it's also the major target of most antibiotics that you're going to dispense throughout your career. But I'm just kind of curious then, if we put cell wall here, no cell wall, what are some other differences then that exist between a human cell and a bacterial cell? And when we ask these questions, really what I'm trying to do is get you to think about how can we target bacteria with small molecules to treat infections. I'm going to stay over here. Yes, sir.
Encapsulated nucleus or membranous nucleus.
Nucleus, all right. So which one has the nucleus?
Human cell.
Good. They're eukaryotes. They have a true nucleus. And inside the nucleus of a human cell, you find the DNA, all the chromosomes. So what's the counterpart then in the bacterial cell?
Just chromosomes.
Yeah, just one chromosome in particular, right? So no nucleus. And the bacterial genome is just one single chromosome, usually a circular chromosome. Good. Tell me more. Ribosomes. Yeah, what do you want to say about the ribosomes before I cut you off? Function-wise, is there any difference between a bacterial ribosome and a mammalian ribosome? No, not really, right? They both accomplish the same feat at the end of the day. Their job is to synthesize proteins. What is unique, though, about these particular ribosomes? Is it that bad? I see your look up there like, what the heck does that say? My handwriting stinks. I know it does. So, which part? This says ribosomes. Not really. I don't know what that says. Ribosomes. and that's in the ADS. Right, so it's a more dense protein is what it boils down to. Right? So the size and the composition then of the ribosomes between the bacterial cell and the mammalian cell actually make a difference. Why is that though? You know, those subtle differences that exist between the bacterial ribosome and the mammalian ribosome allow small molecules to actually have affinity for certain parts of the ribosome and inhibit protein synthesis. And we exploit that with a variety of different antibiotics. Okay, and we'll talk more about those in a little while. All right, so this is off to a good start here. Cell wall, no cell wall, nucleus, ribosomes, what else? Yes. Have plasmids. So, which ones have plasmids? I'm sorry, I didn't. Human cells.
Oh, and bacterial cells.
The bacterial cells have plasmids, and the human cells don't, right? That's what. Yeah, that's what I thought. That's what I heard. Isn't that what you guys heard? You're right. Bacteria. Bacteria are fraught with plasmids. We'll talk about plasmids in a little bit. Plasmids. Plasmids and no plasmids. Sydney, what's a plasmid anyhow?
It's a piece of DNA that you can.
Okay. So from a research and lab perspective, absolutely. Plasmids have done wonders for biotech, for inserting DNA into cells, transfecting cells or transforming cells, depending on what cell type you're using, for the expression of a protein of interest. Okay. But these plasmids are basically small circular sections of DNA that can replicate on their own. Okay. Bacteria are fraught with plasmids. Bacteria can exchange plasmids. Bacteria can pick plasmids up from the environment. So plasmids are not necessarily good for humans. They're not good for antibiotics because they often carry resistance genes. Okay. So bacterial cells have plasmids. Humans do not. There's evidence out there, though, to suggest that humans, in particular patients with cancer, have small circular sections of DNA that encode certain oncogenes, but we're not there yet. We'll talk more about that next semester. Thank you. Tell me more. Is this really the only difference between them and us? Yes, sir.
Bacterial cells can have a flagella.
A flagella. Okay, good. Cilia or flagellum. Okay, so they're modal. They can move around. Right? What do human cells have to sort of stay modal? Do we have a cilia or a flagellum? We have ciliated cells. Yeah, we don't necessarily have a flagellum coming out of our cells, but we have these pseudopodia, you know, these little projections that can come out, especially with your immune cells. So we have something similar, but not necessarily analogous. And that can actually confer some virulence to certain species, right? Their motility and their ability to stick in particular. Okay, others. Yeah. Okay, good. Peptidoglycan. All right. So the peptidoglycan then being the major constituent of the bacterial cell wall. Right? Do bacterial cells have a cell membrane? They have a cell wall. Do they have a cell membrane? I see one person shaking their head yes, a couple, yeah, absolutely. So we both have cell membranes. What's different about our cell membranes relative to a bacterial cell membrane? Are they both made of a phospholipid bilayer? Andy, what do you think? Humans are, absolutely. Yeah, we have a phospholipid bilayer. Bacterial cell membranes are also a phospholipid bilayer, okay? What provides structure, rigidity, and fluidity to our cell membranes? What sort of substance do we put in our cell membranes to stabilize them.
Proteins and cholesterol.
And cholesterol, yeah, in particular cholesterol. So we have sterols in our cell membrane that contribute to the overall structure and function and stability of the cell membrane. The majority of bacteria don't have sterols in their cell membrane. So they both have a cell membrane. We both have a cell membrane. We just don't have lipids, or excuse me, sterols like cholesterol in a bacterial cell membrane. Okay. All right. Other things. What about the organelles? Tell me about the organelles. We have a bunch of different organelles in our cells. We have membrane-bound organelles. Do bacteria have membrane-bound organelles? No, they don't. Okay, so that's another difference. There's some other metabolic features that bacteria possess that we don't necessarily rely on. One of those in particular is this building block for thymidine synthesis. Anybody take multivitamins? Take a multivitamin? I take a multivitamin. I feel like I need another one right now. But one of the key vitamins in your multivitamin is folic acid. Okay. Why do we put folic acid in our multivitamin?
Because we can't make it.
Very good. Olivia jumped right to my punchline. Yeah, we as humans can't synthesize folic acid, so we rely on exogenous sources of folic acid, either from the diet or from supplementation. What about the majority of bacterial cells? They synthesize their folic acid. Okay. And there's probably a bunch of other differences that exist, but this is probably a good enough list for us right now, right? Which leads me to my next question then. The arrangement of bacteria, chains, pairs, etc., visualized after gram stain, can inform decisions on the appropriate empiric therapy. Do you agree with that, Steve? Get a clinical specimen from somebody, you do a quick gram stain, and it comes back as gram-positive oxide clusters. So does that inform your decision on what to put that patient on? Or if it comes back as gram-negative rods, does that tell you something? Absolutely. It's still a very relevant and important technique used in medicine today. In fact, it's probably underutilized. In particular, I'll show you why in a second when we get to this paper. What shapes do bacteria come in? You've already talked about this. There's cocci. There's bacillus. There are pleomorphic bacteria, which kind of take on their own shape. It's neither a cocci, it's neither a bacillus. In particular, bacteria that are atypical, mycoplasma comes to mind. Mycoplasma pneumonia does not have a cell wall. As such, it doesn't conform to any type of specific morphology. It goes however it wants to go. Right? And then there's these other bacteria, Coccobacilli or Coccobacillus, kind of look like either or. So they can't make their minds up. And as suggested on the previous screen, does the organization or the morphology of a bacterial cell matter? Absolutely. That can tell you a lot without doing any deep dive into the molecular aspects of the infection. Okay. Just simply knowing about how the organism gram stains and what its morphology is can tell you a lot about what you might need to do for a patient. And then that comes to this particular paper that was published last year. You know, when you want to poo-poo the concept of gram staining, actually there's a large, well, not a large study, a couple hundred patients were looked at, patients who had a diagnosis of ventilator-associated pneumonia. They have a breathing tube stuck down their trachea, and they're on a machine for ventilation. Patients that were ventilated that were diagnosed with ventilator-associated pneumonia, half of a group got standard of care approaches to treating ventilator-associated pneumonia. The other half provided endotracheal samples that were gram stained. Based on the results of the gram stain, that dictated what type of antibiotic the patient was going to get. Now, what does all this mean? Standard of care based on guidelines from 2016. I don't know, have these been updated since 2016? The best empiric therapy. 2016 guidelines for ventilator-associated pneumonia, MRSA coverage, and anti-pseudomonal coverage. And then we figure out what's the infection, and then we de-escalate thereafter. That's what 100 patients got. The other 100, we gram stained them. If they had gram-positive cocci clusters, we gave the appropriate gram-positive coverage, i.e. MRSA. If they had gram-negative rods stained, we gave them the appropriate anti-pseudomonal coverage. What do you think the results were? How did the standard of care group fare relative to the gram stain group fare? Which one was more efficacious? So it turns out the gram stain group did better. Okay? They had decreased mortality. They had, I can't remember this. They had increased response, 76% versus 71%. Doesn't sound like a lot, but it is still statistically significant. And mortality was decreased. There's decreased mortality in the gram stain group versus the standard of care antibiotic group, 13.6 versus 17.5%. Okay, so do you need to load up everybody with broad spectrum antibiotics at the onset of ventilator-associated pneumonia? Or can you really rely on this old but reliable technique to help inform your decision? I want you to think about one other concept when we look at a paper like this. Gosh, I'll tell you, if you don't have to use a drug like vancomycin in somebody, that's great. We don't want to give people vancomycin if we don't have to. If we don't have to load somebody up with anti-pseudomonal drugs, we don't want to do that. Right? We don't have to, we don't want to. It's great if we can decrease use. Well, the gram stain group proved that. They had decreased use of anti-pseudomonal drugs. They had decreased use of vancomycin. Right? Whether or not this is pervasive out in the real world, I don't know. Dr. Lewis will tell you a little bit about how doing a nasal swab can predict whether or not somebody has MRSA in their lungs when they have pneumonia later on the semester. All right. And then onto the cell wall again too. I'm not going to spend a lot of time on this one. You guys know that gram-positive organisms have this thick peptidoglycan cell wall. You know that a gram-negative organism has a thin peptidoglycan cell wall. And then you know that some bacteria are referred to as atypicals because they completely lack a cell wall, or they have such a thin cell wall that it's not discernible after gram staining techniques. Okay. But I am curious, if you know that a gram-positive organism has a plasma membrane, and then you know it has this thick peptidoglycan cell wall surrounding it, what does a gram-negative cell look like then? So if this is a gram-negative bacterial cell, and this is its circular chromosome, okay? It's not as straightforward as we would like to imagine. So again, this is a gram-negative organism. So again, I want you to understand that when we talk about gram-negative organisms, the architecture is pretty amazing. On the outside of a gram-negative organism, you have this outer membrane. Right? That outer membrane is composed then of a phosphobilipid layer. Inside or in between the outer membrane and then the cell wall is this space called the periplasmic space. Here's the cell wall, which is peptidoglycan in nature, very thin layer of cell wall. And on that cell wall, those stars are meant to represent the targets of your beta-lactam antibiotics. Those are the penicillin binding proteins. Okay. So my question now becomes, if that's a gram-negative organism and you have this outer membrane, what's the outer membrane composed of? What's it made up of? Phospholipid bilayer. Do water-soluble substances diffuse through a phospholipid bilayer very easily? No, they don't. Okay. Lipophilic substances go right through, no problem. Water-soluble substances are going to bounce off. I don't know what you know about beta-lactams just yet, but I'll tell you right now, beta-lactam antibiotics are water-soluble substances. That creates a problem, doesn't it? The target of the beta-lactam is the star, but the stars are surrounded by this outer membrane shield. How do these water-soluble substances, the beta-lactams, get to their target? We have these channels that are embedded in gram-negative organisms only. And these are called porins. Okay. Porins are the entry portal or the gateway for drugs like the beta-lactams, the penicillins, the cephalosporins, and the carbapenems. And why am I mentioning this right now? Well, later on, when we start getting into resistance mechanisms, what you're going to come to discover is that bacteria, especially street bacteria, have figured out how to manipulate or modify their porin structure so that they can squeeze down on them or change the charge in them, which leads to the exclusion of a lot of antibiotics, a lot of beta-lactam antibiotics. So porin modification can lead to the resistance of cephalosporins and carbapenems. Some bacteria can stop making porins altogether. And if the porins are gone, we're in a world of hurt. Because chances are our beta-lactam antibiotic isn't going to get to its target site. This periplasmic space is also interesting because it literally serves as the reservoir, if you will, for this beta-lactamase. Right? It's the reservoir for beta-lactamases. These are enzymes then that the bacteria will use to chop up the beta-lactam antibiotic. We'll consider something like this again in a while. Do we have any questions or concerns about this? Most of it hopefully is somewhat familiar or a little bit of review. Okay. All right. So fill in the blank then. Let's look at A. Change colors here. So from a morphology perspective, how would you describe what you see in A? Coxine clusters. And if I tell you that it seems blue, what do you suppose that is? Gram-positive. Gram-positive, cocci in clusters. And right away, when you hear that, you're automatically thinking what genes? Staph. If I say gram-positive coxine clusters, what that means probably staph, but can you say it's Staph aureus, Staph epidermidis, Staph lugdunensis based on just the description? No, you can't. So you need to do more biochemical tests to figure out the species of staph that you're dealing with. Okay. All right, somebody tell me about B. Black the left side of the room. I'm so sorry. Coxine chains, yeah, absolutely. All right. And if I tell you that they stain positive, crystal violet sticks, now you have gram-positive cocci in chains, and you guys know that that could be a couple things, right? Most commonly, though, what's it going to end up being? Yes, streptococcus. Strep, I agree. Strep. Strep appears as gram-positive cocci in chains. All right. Group A strep, group B strep. All right. What else could it be, though? What other type of virulent organism that has been discussed that's not necessarily a strep appears gram-positive cocci in chains? Enterococcus. Good. Enterococcus. All right. And then what if these were not chains, but they were just gram-positive cocci in pairs? We would call this diplococcus, that gram-stained positive. What do you suppose that is? Yes. Pneumonia, strep pneumonia. Okay. How about if they are gram-negative cocci in pairs? What might that be then? Neisseria, very good. Good. You guys have been studying. That's awesome. Neisseria, gram-negative. What about C? In my opinion, C could be a lot of things. It's a gram-positive bacillus in sort of this boxcar configuration. When you see that, a gram-positive bacillus, at least in a boxcar shape, that could be Corynebacterium. Okay. But I'm more interested in hearing about what you guys think D might be. Chastity, you're probably going to know this because you answered this question last semester, but if you see this corkscrew shape, what could it be? Leptospira, okay? That's one option. What is another organism that is classically described as a spirochete and has this corkscrew appearance? Leptospira is one particular organism that is classified as a spirochete. There's a surge of this infection going on in the United States right now. There's an increased incidence of neonatal infections secondary to this particular organism. There's a shortage of bacillin LA, making this difficult to treat, not because the organism is resistant, we just don't have the resources to treat it. We are importing this from France. The drug, that is, not the infection. Don't need to import it. It's already here. Spreading every weekend. Do you know what Treponema pallidum is? Have you heard of that? Has anybody heard of Treponema? Have you heard of syphilis? Yes. All right, so syphilis is caused by the spirochete Treponema pallidum. Treponema pallidum, like Leptospira, has this corkscrew appearance. And then this last picture here on the bottom, I mean, it's a terrible picture. I don't even know why I kept it in there, but I did. If that gram stain's negative, you know it's a gram-negative rod. This one has a single flagellum. This could be a variety of different organisms. It could be cholera, it could be E. coli, maybe it's a Klebsiella, I don't know. But when you see a gram-negative rod show up on a gram stain, you don't know what it is. You need more biochemical tests to identify the organism. But what that does do, though, is it does inform your decision on what particular antibiotic the patient should be on to address the presence of that organism. Okay, questions about this? All right. Gram-negative organisms possess a substance called lipid A, which can induce symptoms of shock, septic shock. You think that's true or false? Have you heard of lipopolysaccharide, LPS? Yes. One of the constituents of LPS is lipid A. It's a fatty acid chain that helps to embed lipid A or lipopolysaccharide into the or the outer membrane of gram-negative organisms. And then gram-positive organisms have something called teichoic acid, which can also induce symptoms of shock, septic shock. And that is true as well. Teichoic acid is a substance in gram-positive organisms that often lends to a stickiness nature. It aids in adherence. Okay, so LPS. LPS is essentially what we refer to as an endotoxin. Okay, so gram-negative organisms have that outer membrane, and that outer membrane has embedded in it lipopolysaccharides. And those lipopolysaccharides stay in place. However, when that gram-negative organism dies, secondary to the immune system or secondary to antibiotic presence, that LPS gets shed, it gets released into the system, into the patient. And if there's enough of it, what it can do is it can start to interact with components of your innate immune system. Right? Your innate immune system, one portion of which is the toll-like receptors, recognizes the presence of peptidoglycan. It recognizes the presence of double-stranded RNA. It recognizes the presence of lipid A. And it says, okay, we got something going on here, because the toll-like receptors are pathogen-associated pattern recognition receptors. They see peptidoglycan. They know that it doesn't belong. They sense lipid A, and they know that's a foreign substance. The toll-like receptors know that we don't make double-strand RNA. If we do, it gets chopped up really, really quick in the cell. All right. What that does then is when this substance, this lipid A from LPS binds to a toll-like receptor, it starts to stimulate then the production of cytokines, trying to ramp up the immune system to fight an infection. Okay. LPS, again, lipid A, a core polysaccharide, and then the O antigen, the outer portion, right, of LPS. The O antigen in gram-negative bacteria is actually how we serotype certain bacteria. So you look at all the different E. coli that are out there, they're not all the same. They all have different O antigens, and that's how we identify this serotype to that serotype to that serotype. This is the bad part of LPS, though, lipid A. Right? And when you blow it up, you can see here, lipid A has got all of these fatty acid tails that embed into the outer membrane. Lipid A can actually interact with TLR4 to induce an immune response. And if that immune response is so exaggerated, it can actually lead to shock. And most of the time, it's going to be septic shock. I'll just put here for an example. There was a drug that was being studied years and years ago. It was called Eritorin. It's made by, it was marketed or studied by a company called Eisai. It was meant to be a TLR4 antagonist. So patients that presented with signs and symptoms of shock that were receiving appropriate antibiotic therapy and other best supportive care measures were enrolled in this study to see if this particular antagonist, Eritorin, could block lipid A from binding to the toll-like receptors to prevent then this exaggerated response. And well, the drug never got approved, but conceptually speaking, it sounded like a really cool idea. They studied it for a while. The mortality didn't fare out so well, so it got dropped. From the gram-positive side, again, too, gram-positive organisms. Here's a bonus question. Can anybody tell me, you don't have to tell me now, there's at least one gram-positive organism that makes LPS. Tell me what that is when you have a chance, right? But the preponderance of gram-positive cells don't make LPS. Gram-positive cells do make teichoic acid. Again, teichoic acid is this substance that bacteria, gram-positive organisms use for adherence. When gram-positive organisms succumb to the presence of antibiotics or the immune system, the teichoic acid can get shed. And teichoic acid, much like lipid A, can bind to toll-like receptors to induce symptoms of shock, all right? blood vessels dilate, capillaries get very leaky, immune system goes kaput, hypovolemia, you get hypotension, bad things happen. We'll talk about that later on this semester. Okay. Shifting gears, the cell membrane and then the cytosol. We talked about the cell membrane. We already said that the cell membrane is composed of a phospholipid bilayer. And we also said that the majority of bacteria lack sterols in their cell membrane. Why? They don't need them. They don't need sterols in their cell membrane because they have a cell wall that gives them shape and protection, right? And it protects them from osmotic pressure, so they don't just pop. The exception there again, too, is mycoplasma. Why might mycoplasma embed sterols in its cell membrane? Very good. No cell wall. So it needs something else to keep it from going kaput. We talked about the ribosomes. We talked about the differences between mammalian ribosomes and human ribosomes. Again, 70S is what you get when you put a 50 and a 30 ribosome together in a bacterial cell. 40 and 60 in a mammalian cell gives you the 80S. And then we said the cytosol is where you're going to find the bacterial genome. All right. The genome itself is typically composed of one circular chromosome, and that circular chromosome that circular chromosome houses then all the genes that the bacterial cell needs for its replication and survival. Okay. I just think it's interesting, nothing more than that, but Vibrio and Borrelia kind of go against the grain of what's normal. Most bacteria have a circular chromosome, just one. Vibrio ends up having a large circular chromosome and a small circular chromosome. I don't know what the evolutionary advantage is there. And then Borrelia are kind of interesting. Borrelia burgdorferi. Can anybody tell me what disease is caused by Borrelia burgdorferi? Lyme disease. Very good. So Lyme disease is caused by Borrelia. Borrelia does not have a circular chromosome. In fact, it's got a linear chromosome. Again, we don't know how or why, what's the evolutionary advantage there is. I don't even think they're really sure how that chromosome gets replicated, but nonetheless, it is what it is. And then we talked about plasmids at the beginning of our talk. Extrachromosomal, circular, double-stranded DNA, okay? Plasmids. Plasmids can go from one cell to the other. They can be exchanged pretty easily. They can replicate and get expressed independent of what's going on with the chromosome, all right? And why are plasmids so relevant or so interesting from a microbiology perspective? Well, this is just a short list of things that you can find encoded on plasmids. First and foremost, lactamases, beta-lactamase enzymes. This is probably one of the things that we're most concerned about today. We'll talk about beta-lactamases as a resistance mechanism to penicillin cephalosporins and carbapenems. Okay? And we'll talk about the classification of beta-lactamases. Most beta-lactamases are encoded on plasmids. Some of them are encoded on chromosomes as well. Exotoxins, not endotoxins, but exotoxins. A lot of organisms will make and secrete exotoxins, and these exotoxins that do damage to patients in particular are encoded on on plasmids. Attachment and motility proteins, pili and fimbriae, encoded on plasmids. We used to rely on UV light as a mechanism of sterilization, coming to find that UV light might not be as beneficial as once thought. And maybe that's because bacteria have adopted or acquired genes that encode proteins which enable them to repair their DNA after UV exposure. UV exposure can actually break apart DNA, but if the bacteria has a gene that codes a protein to put it back together, that's going to be a problem. and then a constellation of other resistance genes that we'll talk about, in particular the TET genes. The TET genes, of which I don't know how many there are now, just tons and tons of TET genes. These encode for efflux pumps. The majority are efflux pumps that bacteria can get a hold of to pump out antibiotics from the inside of a cell. The antibiotic isn't inside the cell where it needs to be because it got pumped out. It doesn't do us any good. All right, we'll pause here for a moment, take a break, come back at, I wish it was 10:50. You do too, I bet. But we'll come back at 10 and pick it up. All right, let's pick it back up. Just reviewing our bacteria. Some bacteria have this thicky, gooey substance that coats them. It's called a capsule. And the capsule itself is a polysaccharide coating. And sometimes this polysaccharide coating can be quite pronounced. It can be hypermucoid is what we like to refer to it when it's very, very thick and pronounced, as you see up on this image here. Regardless, this polysaccharide coating serves as a protective layer for many bacteria, encapsulated organisms. And we actually can use this capsular material to serotype bacteria as well. So when you look at Streptococcus pneumoniae, there are over 90 different serotypes of strep pneumo, and we serotype them based on the composition of this polysaccharide capsule. Again, too, the polysaccharide capsule, it's a virulence factor. It makes bacteria much more difficult for the immune system to eradicate. It prevents phagocytosis. It protects the bacteria. So it can be difficult to eradicate such organisms in the absence of a little help, maybe some help from antibiotics. It also aids in attachment as well. The more interesting aspect, though, of capsular material is what it can provide us with, from a protective measure. What can we do with a bacterial capsule component? What can it actually provide us with? Yeah, actually, you could probably take that capsular material, bits and pieces of it, that's what's conserved. You might be able to raise monoclonal antibodies against it, okay? And then that could go on and be used as sort of a third line salvage therapy in certain individuals. I'll have to look to see. I'm not aware of any monoclonals being developed against the polysaccharide. It's interesting to look at, okay? But I did mention antibodies. Well, when's the last time you guys gave a vaccine? Have you ever given pneumovax vaccine? Yeah. You look at what's in that pneumococcal vaccine. It's a multivalent polysaccharide. It's typically derived from polysaccharide capsule of strep pneumo. So we're actually immunizing patients against the capsule itself to raise antibodies. Okay? So we can use the capsular material for vaccination purposes. Also do that with meningitis. All right. That's all I'm going to say about our quick little bacterial review. Again, too, nothing really heavy from that content. Just want to kind of bring everybody up to a level playing field. And now what we can do is we can start diving in, talking about some molecular aspects of bacteria that's going to help then hone in our understanding of antibiotics. All right, so we're going to spend some time talking about molecular targets of various antibiotics. And we're going to look at DNA synthesis. We're going to look at RNA synthesis and protein synthesis. DNA precursor production very, very briefly. And then we'll finish up with cell wall synthesis. All right, but before we get to that, I've handed out 2 pages thus far today. The first one is a rendition of this page. Right now, I don't know how much of this you could fill in on your own. I'm not really concerned about it at this point in a couple of weeks. So what I would do is hand you a blank piece of paper and have you go around and fill in all different drugs in those gray boxes. We'll fill these in as we go. I understand that everybody comes into the class with varying levels of experience. And as we said on week 1, ID is kind of like learning a second and third language. So this is kind of like our vocab cheat sheet, if you will. All right. Is this class complicated? Yeah, it's hard. I know it's hard. There's a lot of stuff going on. All these dang bacteria, all these drugs, and then we got to figure out how we match them up and pair them up. There's a lot of classes of drugs. You know what? There's a lot of classes, and then there's a lot of drugs in certain classes. But what's interesting is that despite all of these drugs in some of these classes, we don't use them all, okay? In fact, there are critical drugs that we need to be very familiar with, the common drugs to treat common infections. And that's what I'm really going to emphasize. We will spend some time talking about special circumstances where you need to employ specific drugs for difficult to treat organisms. But for the majority of the content, here's a common infection, here's the common antibiotic. That's what you need to have crystallized in your mind, right? But you may ask yourself, if we have all of these drugs in all of these different classes, why aren't we using them? And there's a variety of reasons for that. Efficacy is probably one of the main reasons. Efficacy has continued to plummet in the recent years due to resistance. Bacteria, I mean, they mutate their DNA so easily with almost every single replication. It's not surprising that they arrive at some type of resistance mechanism by chance, all right? And then selective pressures. Again, too, when that one strain, that one bacterial cell has a mutation, and then you put an antibiotic on board, that one cell gets selected out from the rest because it's resistant to the antibiotic, all right? So selective pressures. A lot of the older drugs that are no longer in use are relatively toxic, okay? So we can't do more harm than good by giving an antibiotic. Although we get faced with certain circumstances where you have to take a chance. If the toxic antibiotic is the only one that has activity against the infection, well, you're going to die from the infection, or you're going to have some renal deficit or renal impairment or renal damage secondary to the antibiotic. Take your pick, okay? But toxicity has a lot to do with why we don't use certain antibiotics anymore. And we've come out a long way with regard to antibiotic development. So from a pharmacokinetic perspective, you know, we don't want to have to give drugs every 4 hours, all right? We don't want to have to give drugs every 6 hours, whether that's at home or in the hospital. Every 4 hours, that's a huge strain on nursing staff. To go in there and hang a new bag every 4 hours takes a lot of time and resources. 6 hours as well. And we have antibiotics out there these days that we only have to give 1 dose of, and it lasts a week. We have antifungals that can be given once weekly. All you need is 2 doses to take care of the infection. So from a PK perspective, we keep coming out with clever ways to circumvent the need for multiple administrations. And then spectrum of activity. You know, when you look at spectrum of activity, most of the new antibiotics that have come to market in the last 10 years or so aren't necessarily what I would consider broad spectrum antibiotics. They're actually somewhat narrow spectrum, focusing in on 1 or 2 difficult to treat organisms. And I'll show you what I mean in a couple slides. You may have mentioned this or not, I don't know. We use the term antibiotic, we use the term anti-infective, we use those interchangeably. The classical definition, again, of an antibiotic is something that's of natural production, naturally derived, right? So an antibiotic, antibiosis, anti-growth, right? That's something that's made in Mother Nature's lab, if you will. And antibiotics, again, the major source, the major inspiration for the majority of antibiotics that we still use today. Okay. These antibiotics can be fungal metabolites, like the penicillium mold that we're all familiar with. They could actually be bacterial metabolites, right? We have bacteria out there that synthesize and secrete metabolites that kill off other bacteria, okay? So think about that for a second. You have a bacterial cell that's synthesizing and then secreting into the environment a substance that kills bacteria. That creates more real estate for that particular bacteria. It creates more resources for that particular bacteria that made that metabolite. But why does that bacterial cell not fall victim to its own metabolite? Why doesn't it destroy itself in the presence of its own metabolite? Everybody with me? Understand the question? Do I need to rephrase it differently? Say it differently? Here's a bacterial cell, and this bacterial cell is pushing out the stars. Then here's a different bacterial cell. But this bacterial cell doesn't like the stars. And as a result, it dies, leading to more space and resources for the red bacteria. But why does the star, bless you, not circle back and kill the red bacteria, just the green bacteria? They would have resistance. Yeah, there's resistance already in place. Resistance mechanism that exists in that red bacterial cell that protects it from its own toxic substance. Now, the bad news is that what if this resistance mechanism that's in place in the red bacteria somehow makes its way into the green bacteria, which then somehow makes its way into other bacteria? That becomes a problem, all right? That's how you get this spread, horizontal spread of resistance mechanisms. Okay. We typically don't take that bacterial metabolite and use it as is. That rarely happens these days. But what we can do is we can take a portion of that metabolite, we can chop off a piece here or add a piece there. And what we're afforded then is something that we refer to as a semi-synthetic antibiotic. Most of the drugs, most of the beta-lactams that we use today are semi-synthetic. We keep the cephalosporin core, we keep the penicillin pharmacophore, or the carbapenem pharmacophore, and then we might add to it, okay? That's a semi-synthetic antibiotic. Antibacterial. Antibacterial is the term we use to describe a substance that was purely synthetic. It was made in the lab by scientists without Mother Nature's involvement, okay? The oxazolate dinols, like linazolate, the fluoroquinolones, Cipro, Levo, right? Those are antibacterials. I'm going to use those terms interchangeably. I'm going to say antibiotic when I meant antibacterial. I'm going to say antibacterial when I meant antibiotic. I want us to be comfortable with calling them whatever we want, okay? Because here it doesn't really matter. What does matter, though, is this last part. If you're in the pharmaceutical industry, do you want to be messing around with antibiotics? Do you want to invest a lot of time and resources into antibiotic development? Probably not. Why not? Yeah. Kristen. It's a one and done. Oh my gosh, yeah, pretty much. Use it and lose it, right? You use it, you're gonna lose it. That's the problem with antibiotics. You come out with a new antibiotic today that's never been used in a clinical setting, and somewhere out there in the world, there's already a bacterial species that has a resistance mechanism to it. And if it's not already in existence, it only takes a little bit of exposure to that antibiotic to develop some type of resistance mechanism. We've not arrived at that holy grail where the bacteria have no ability to develop resistance. They're going to find a way. They evolve too fast. They replicate too fast. Right? you want to make a drug that somebody's going to take for 5 to 10 days, or do you want to make a drug that somebody's going to take daily for the rest of their life? That's the problem. I give the government a little bit of credit. They did have initiatives to get the pharmaceutical industry to dump more time into antibiotic development. I think it was 20 by 2020. It was a campaign that was introduced by the government to come out with so many new antibiotics by 2020. And they didn't do too bad of a job. But, we're still lagging as far as I'm concerned. You can see from a timeline when all these various and sundry antibiotics were introduced into the clinic. I mean, sulfonamides came around and started to really be used between World War I and World War II. And then penicillin was discovered in 1941, but it didn't really start to get used until the mid to late 40s. Production of penicillin was very, very difficult back in the day. They just didn't have, the ability to grow up enough mold to generate enough metabolite to procure it for using it as an antibiotic. It took a long time to figure that out. The aminoglycosides, again, from a Streptomyces species, those were identified in the 40s. You can see this was a very, very exciting time for antibiotic discovery and development. And then you hit a lag. I guess the 70s were pretty fun. I don't know, but not much going on there. And then in 2000, that's when linezolid hit the market. And then in 2003, lipopeptides, specifically daptomycin or cubicin, came to market. So antibiotic development waxes and wanes. And then when you look here, daptomycin in 2003, tigecycline, you look over the last 20 years, with the exception, I'll cross them out here. You get rid of this one. The majority of those antibiotics listed above this line are intended to treat what type of infection do you think? Telavancin, tedezolid, dalbavancin, ceftaroline, daptomycin. Remember, I said, in my opinion, the drugs that have been discovered as of late are not broad spectrum, they're relatively narrow spectrum. So if we're using that lens, what organism do you think the drugs listed at the top there are really focused in on? Yeah, MRSA, MRSA in particular. Can they be used for other organisms? Yeah, MRSA is probably the most common. Some of these are used for VRE, vancomycin-resistant enterococci, but difficult to treat gram-positive organisms represent the preponderance of indications for these top agents. How about down here? We'll take doripenem and throw Dory down here as well. Ceftolozane, tazobactam, ceftazidime, avibactam, meropenem, vaborbactam, and so on, cefiderocol. What are these targeting? You betcha. Difficult to treat gram-negative organisms with multiple resistance mechanisms. Unfortunately, these difficult to treat organisms don't harbor just one resistance mechanism or one resistance enzyme. They probably have multiple resistance mechanisms, making them, well, as the name implies, difficult to treat. So we keep going on and on and on. If you go to clinicaltrials.gov and type in antibiotic and infection in your search term, you'll get over 7,200 hits. Are all of those interventional studies? No, certainly not. But a lot of them are, and they're looking at new antibiotics, new combinations to try and combat these resistance mechanisms, right? That's the big problem here. We don't want to develop drugs. We don't want to pour a lot of time and effort into drugs that are going to be toxic. So those get weeded out pretty quick, okay? And then what blows my mind is the way that we can manipulate a structure to keep it around in the body for prolonged periods of time. Prolonged periods of time. Not a couple hours, we're talking days and a week. It's pretty amazing. So you guys are going to see new antibiotics come to market over the course of your careers. You're going to see new therapies come to market. And it's my opinion that if you have a good handle on what bacteria rely on from a metabolic and reproductive perspective, if you understand the machinery that's involved when a new drug comes to market, you should be able to assimilate that in your mind pretty easily if you're pretty comfortable with what's going on inside a bacterial cell. Okay? Right. This was published last year. There's a drug called tebipenem that's being developed. Tebipenem for complicated urinary tract infections. And when you look at the structure of tebipenem up in the right, maybe not today, but in a couple of weeks, you'll quickly recognize that as a carbapenem. Okay? Okay, fine. Yeah, we got carbapenems on the market already. What's so cool about this one? Well, what's cool about this one is that this one's being developed as an oral carbapenem. We don't have an oral carbapenem right now. But boy, if we did, bring the patient into the hospital, identify what type of organism is growing in their urinary tract, get them stabilized and recovering on IV, and then get them out of the hospital on an oral agent, right? An oral agent that is going to afford you reliable coverage for the offending organism, right? Do we want to just put everybody on a carbapenem? No. But what's the drug of choice for ESBL producing Enterobacteriaceae? Carbapenem. Okay. So it's a prodrug that gets orally absorbed. And there is a lot of traction going on right now in the clinical setting with regard to developing oral agents, novel oral beta-lactamase inhibitors in particular, that can inactivate some of these nasty enzymes that are responsible for breaking down third-generation cephalosporins. Some of these being developed can restore activity of a third-generation cephalosporin to cover organisms that are producing ESBLs and to organisms that produce cephalosporinases. So it'll be exciting to see what comes forward in the next couple of years. Okay. All right, questions or concerns? How are we doing? Everybody with me? So? No? Yeah. All right. Let's talk about some classes and their targets here. And as we go through this, again, too, I want you to think about what's the role or the function of that particular target in a bacterial cell. Importantly, is that target unique to a bacterial cell, or is it also expressed in the mammalian host? If we inhibit that particular target, what's going to happen to the bacteria? Or, more importantly, what do bacteria do to get around the presence of that organism, or excuse me, of that molecule? Okay. So first off, we're going to talk about DNA and RNA synthesis. Okay. And again, too, I think I've mentioned this in the past. I don't remember. But again, too, a bacterial cell itself, on average, is very, very small, 1 micron. But the circular chromosome that's packed up inside that bacterial cell is relatively large from a length perspective. 1 micron versus a couple millimeters. 1 millimeter. You cut that circular chromosome in half and stretch it out, it's much longer than the diameter of the bacterial cell itself. So that creates a problem for bacteria. You have a bacterial cell, but then all bunched up inside that bacterial cell is this long circular chromosome. How does it replicate that chromosome? Number one. And number two, how does it access the information in that chromosome to make proteins and to replicate? Because it sounds like it's a pretty complicated process if you ask me, just looking at the numbers. And it turns out bacteria have these enzymes that they use to cut, open up, gain access to, and then reseal for the purposes of replication and expression. Right? There's 2 enzymes in particular that we need to be familiar with, DNA gyrase and then something called topoisomerase 4. And these 2 enzymes are type 2 topoisomerases. I don't know if that term is familiar to you from your cell biology days, but a topoisomerase is classified by the number of nicks that it will create in a strand of DNA. If it cuts on both sides, we refer to that as a type 2 topoisomerase. If it induces one nick, that's a type 1 topoisomerase. These two bacterial enzymes are type 2 topoisomerases. What that means is that the enzyme will grab onto a section of DNA, it will cut that DNA and pull it apart, allow some type of metabolism to occur, and then importantly, it will put those two pieces of DNA back together and reseal them. When bacteria are replicating or expressing their DNA, you have this replication fork, okay? And remember, this is double-strand DNA, right? It's twisting, it's turning, and if you grab on here, and you grab on here, and you start pulling apart that DNA for the purposes of replicating it, what you're going to do is you're going to start inducing a lot of torsional strain on that replication fork. And at some point, it's going to get bunched up. It's like giving the DNA a big wedgie is what it boils down to, okay? And we don't want that if you're a bacterial cell. So what the bacterial cell does is it has a way to cut and unwind it and reset it and allow the process to continue. And then it can cut again, and then it can cut again and again and again to prevent this twisting phenomenon. And what that does is that allows this process to go. And in some bacterial cells, it goes very, very quickly. E. coli replicates very, very fast. Right? The name of the enzyme that accomplishes this feat is the DNA gyrase enzyme. After replication is complete, you have one circular chromosome, which we'll call the paternal chromosome. And then you have the daughter chromosome. So when you have a circular chromosome that gets replicated, at the end of replication, you end up with a chain, all right, 2 links. And these links need to come apart so that when binary fission occurs, you have a chromosome here and a chromosome there in the respective bacteria. But how does that happen? What enzymes do bacteria have to cut, separate, and then reseal such that that chromosome can then find its home? One of the oh well, that's gone. One of the functions of topoisomerase 4 is to actually do that. It will cut and reseal the bacterial chromosome such that you have one chromosome in the daughter cell and then one chromosome in the parental cell. And then after the end of binary fission, you have 2. Now think about this for a second. If you were inhibiting some of the function of gyrase or topo 4, you can actually induce a lethal effect in bacteria. So I want you to pause and think about this for a second. If you inhibit the activity of gyrase or topo 4 completely, will that have an effect on bacteria? Maybe, maybe not. Remember, gyrase needs to unwind and relax that DNA a little bit. Right? Topoisomerase 4 needs to cut and then reseal. If it doesn't cut, it can't separate. Okay? So by virtue of doing that, that might have an effect. But the real effect of drugs that inhibit gyrase and topo 4 comes from their ability to prevent the resealing activity of these respective enzymes. Okay? Go back to DNA gyrase. DNA gyrase is a type 2 topoisomerase enzyme. Okay? This tetrameric protein is grabbing onto 2 strands, right, of DNA, right, opposite sides. It's cutting, separating, and then resealing. when you put a fluoroquinolone antibiotic, like ciprofloxacin, for example, on board, ciprofloxacin is not going to inhibit the cutting aspect. What it's going to do is it's going to inhibit the resealing aspect of the bacterial DNA gyrus. It prevents the religation or the resealing of the bacterial genome. And what that results in, you guys, then is the accumulation of all of these breaks along the bacterial chromosome. That bacterial chromosome is going to shatter, and that's going to induce a bactericidal effect in your target organism. Much like is shown here. You see how all that chromosome looks fragmented? That's really what we're trying to accomplish with the use of fluoroquinolones. We don't have a gyrase enzyme. Right? We get around this coiling issue a little bit differently because we have linear chromosomes. Right? So we don't worry about that so much. We do have a topoisomerase 4 enzyme, but our topo 4 is insensitive to clinical concentrations of fluoroquinolones. So we don't worry about giving individuals who have an infection a fluoroquinolone and having it inhibit our topo 4. As such, what we can do is we can selectively inhibit DNA gyrase and bacterial topo 4 for the purposes of treating an infection. I'm going to go over these pictures really quick. I think what I really want you to see here is that the enzyme itself is a heterotetramer. There's par A and par C, and then GYRA and GYRC. Don't worry about that right now. We'll get into that next week. But this tetrameric protein, this enzyme, wraps itself around the DNA, and it uses a tyrosine residue as a nucleophile to attack the phosphodiester bond in the DNA. And as it does so, what it does is it creates these breaks here, and then the other one is back there. You can't really see it. That's where they come apart. The presence of the drug will prevent them from coming back together. The presence of the drug prevents the resealing action. And I want you to think about this for a second. This is what the chromosome looked like before the introduction of the fluoroquinolone. And then after the fluoroquinolone is on board, you have all of these little nicks, all of these double-strand breaks, which is lethal then to the bacterial cell. Okay, come on. And we're locked up. So, how was your weekend? Good. Technology. I mean, I don't even really think I'm that technologically challenged, but I guess I am. So, I can't even close it. Come on, man. There we go. Maybe not. Finally. Maybe I am challenged. Oh boy, it's gonna be a long semester. What class of enzymes or class of drugs does this affect? What class of drug, exerts this mechanism of action in bacterial cells? This is where the fluoroquinolones come into play. The overall effect on bacteria is largely bactericidal. And there's 3 or 4 important fluoroquinolones that you need to be familiar with, but they are really quick. Yep. You guys have already heard of those, so I'm not going to spend too much time on those. All right, good. We'll talk more about the structure activity of fluoroquinolones, the mechanism of action, resistance mechanisms we will mention, and side effects as well, the fluoroquinolones as we get into next week. Nucleic acid precursor synthesis. Yeah, I think we'll hold off on that. Okay, nucleic acids. Again, two. I mentioned this at the onset of today. The majority of bacteria need to synthesize their folic acid. All right. They need to synthesize their folic acid. And when they synthesize folic acid in a bacterial cell does the same thing as folic acid in a mammalian cell. It gets metabolized and then ultimately turns into a one carbon donor for the synthesis of thymidine from uridine. Okay, so remember, uridine and uracil is found in RNA. Thymidine is what we put in DNA. All right, we make thymidine from uridine, and we do that by metabolizing folic acid. We get our folic acid from our diet. Bacteria synthesize folic acid. So it's reasonable to think that if you can identify a small molecule or small molecules that interfere with the synthesis of folic acid in a bacterial cell, you can suppress folic acid production. And if you suppress folic acid production in a bacterial cell, you'll inhibit DNA synthesis. Okay. There are two interesting enzymes that bacteria have that are responsible for facilitating this folic acid synthesis and then this downstream metabolism into this one carbon donor. The first one I'll make mention of is something called dihydrotoroate synthase. Dihydrotoroate synthase. This basically takes para-aminobenzoic acid and dihydrotoroate diphosphate, smashes them together in its enzyme active site, and makes metabolites that ultimately form folic acid. Dihydrofolate reductase, we'll talk about that in a second. If you inhibit this enzyme, along with that enzyme in particular, you can largely induce a bactericidal effect in susceptible organisms. Okay. Like I said, do we use these enzymes? Not the dihydrotoroate synthase. We don't need that enzyme because we take our folic acid from our diet. This other enzyme that I made mention of, dihydrofolate reductase, we do have a dihydrofolate reductase, which we do use, right, to make a one carbon donor for our own thymidine synthesis. But I will tell you that the drug that inhibits this pathway in bacteria doesn't have any effect on our mammalian enzyme. You know, the differences in amino acid structure and composition of the enzyme itself are just different enough to where the drug doesn't impact our process. So in susceptible bacteria, we can accomplish this selective toxicity. Here is that dihydrotoroate diphosphate that I made mention of. Bacteria will synthesize this precursor. And then in the context of the synthase active site, it will combine dihydrotoroate diphosphate with para-aminobenzoic acid. Okay. The amino group at the para position will actually attack this methylene carbon between the ring system and the diphosphate. It'll kick off the diphosphate as a leaving group. And what you're left with, guys, is this intermediate dihydrotoroic acid. And then this carboxylic acid on this intermediate is going to get joined with glutamic acid. And that's how bacteria, in a nutshell, make folic acid. Okay. And then folic acid will get reduced by dihydrofolate reductase into a metabolite called tetrahydrofolate. Tetrahydrofolate goes on to get metabolized a couple more times, and ultimately what we get is a one carbon donor that shuttles that carbon to uridine. We'll talk more about that after a while, but what I want to impress upon you is that the sulfa drugs, in particular sulfamethoxazole. Sulfamethoxazole, one of the ingredients in Bactrim, works to inhibit this enzyme to prevent the formation of that substance. All right, so sulfamethoxazole suppresses the formation of dihydrotoroic acid. If we don't make dihydrotoroic acid, we don't make folic acid. We don't make folic acid. We don't make tetrahydrofolic acid. You don't make thymidine. Sulfamethoxazole's counterpart is trimethoprim. And trimethoprim is an inhibitor of bacterial dihydrofolate reductase. Dihydrofolate reductase is responsible for reducing the ring in dihydrofolic acid to tetrahydrofolic acid. It actually just shuttles 2 protons across the ring and reduces it. But nonetheless, if you inhibit dihydrofolate reductase, you don't make tetrahydrofolic acid, and then you don't go on to make thymidine again. So you induce thymidine starvation in susceptible bacteria. And here's an image of the dihydrotoroate synthase. And then pictured in here is an inhibitor, sulfamethoxazole. Okay. The clinically relevant combination is Bactrim. We'll talk more about the structure and the relevance of the charge of Bactrim later on. All right. On to protein synthesis. All right, so again, protein synthesis. Bacteria, just like mammalians, central dogma of molecular biology. Right? DNA to RNA, RNA to protein. They follow the same pathway we do, okay? But what's different, again, as was mentioned earlier, just the composition of the ribosome, okay? It's a little bit different. Structurally, it's still made up of protein and ribosomal RNA, just like mammalians, but the topology and the architecture is a little bit different, such that certain substances out there that we can get from synthesis, from natural sources, have the ability to interfere with bacterial protein synthesis. Again, to the large subunit, you guys know this, 50S, the small subunit is the 30S. When they come together, you get a 70S ribosome. And then that 70S ribosome has got a couple interesting areas located inside it once it comes together. All right. There's the exit site. There's the peptidyl transferase site. And then this is the amino acyl transfer RNA site. Okay. And then what's missing on this particular image is the actual messenger RNA. Right. So again, too, you guys know this, right? I feel like I'm just wasting time, but a transfer RNA with an amino acid will come into the A site and bind, and then another transfer RNA with an amino acid will come in, and so on and so forth. And ultimately, what you get is a growing peptide chain. And then the amino group on this particular amino acid is actually going to attack that particular ester bond that's connecting the polypeptide to the transfer RNA in the P site and grab onto it. And then this particular transfer RNA is going to exit through the exit site. And it just keeps going on and on and on until a stop codon is reached. Well, what if we identify a small molecule that has the ability to bind in this imaginary A site of the ribosome? What if we have, if this is the A site, amino acyl transfer RNA site, this is the P site, and then this is the E site. And my iPad is a small molecule blocking that A site. And then I'm a transfer RNA with an amino acid. I want to come in here and I want to sit down in this A site, and then I want to attack an ester bond that's connecting a polypeptide in the P site so that I can grow by one more amino acid. Okay? That's what I want to do. But if something's blocking my ability to plant it in the A site, I can't get here because matter can't occupy the same space. So what this can do is or prevent protein synthesis if we know what molecules will do this. Okay? And there's a couple of them that do that. The tetracyclines in particular bind to the A site in the ribosome to prevent protein synthesis. If you look down here, agents that bind to the 30S aspect of the A site include tetracyclines, doxycycline, minocycline, and then classical tetracycline, which I don't really think is used all that often, if ever anymore. These drugs physically plant it in the A site, right, on the 30S side, and prevent incoming amino acyl transfer RNAs. Okay, protein synthesis slows down. There's another class of antibiotics referred to as the aminoglycosides. The aminoglycosides, gentamicin, tobramycin, amikacin, plasmamycin, maybe. Right? These work a little bit differently. They do bind into a similar area of the bacterial ribosome, but rather than creating sterics such that an incoming amino acyl transfer RNA can't bind, what they do is they actually interfere with the ribosome's ability to correctly read the messenger RNA. Okay? So when an aminoglycoside is bound to the ribosomal RNA in the bacterial ribosome, what's going to happen is that you get a skipping phenomenon. You're supposed to read a codon, and then another codon, and then another codon sequentially. But when the aminoglycoside is on board, a codon will get read. A codon will get read. We'll skip. We'll skip. A codon will get read. We skip. A codon will get read. And what this leads to then is the production of what we refer to as aberrant proteins, nonsense proteins. These nonsense proteins that get produced, or missense proteins that get produced, actually go on and cause damage to the bacterial cell itself. Right? So we're not necessarily inhibiting protein synthesis completely with an aminoglycoside, but what we are doing is tricking the bacterial cell into producing something that's going to cause damage to itself. It's going to make a protein that has the ability to go out and then interact with the cell membrane and maybe poke holes in it or do damage to it. And that induces then a bactericidal effect in susceptible organisms. A nonsense protein or an incomplete protein, that's what we get when we have tetracyclines and aminoglycosides on board. Okay. This is all on the smaller subunit. If you go back to this aspect here, what site is this? This is the P site. What happens in the P site? The amino acid in the A site attacks the growing peptide chain in the P site, right? So there's got to be an interaction there. What if we have small molecules that bind in this P site and interfere with that particular nucleophilic attack? What if we can identify a small molecule that's going to prevent the amino acyl transfer RNA, the amino acid in the A site, from attacking the peptidyl chain? If we can do that, what we will do is shut down protein synthesis. We'll inhibit protein synthesis. And there's a couple drugs out there that actually work to bind in that P site. Right? They interact with ribosomal RNA. They establish a nice interaction with the ribosome, and they prevent then peptidyltransferase reactions from occurring. In particular, these are the macrolide antibiotics. The macrolides, azithromycin, clarithromycin, and then historically speaking, erythromycin. Again, you get incomplete proteins that do nothing then for the bacterial cell. What this does is it can slow down then bacterial replication, kind of puts the bacteria on pause. And what that does then is it allows your immune system to catch up. So hopefully reconstitute the immune system, let the immune system start to eradicate all of those organisms, and get a clinical cure, hopefully. So how does a macrolide, or how does a tetracycline bind selectively to a bacterial ribosome, but not the human ribosome? Again, too, if you were to look at the crystal structures of these drugs bound to their targets, you would see some beautiful interactions that exist between the drug and the ribosomal RNA. It's fascinating. I'll show you a couple pictures. But these interactions don't exist in the human ribosome. They're not there. You have to give gobs and gobs and gobs of antibiotics in order to shut down mammalian protein synthesis. So again, this lends back to this concept of selective toxicity. You can selectively target and kill bacteria through these interactions. Oh, geez. Cell wall. I always run out of time at the cell wall, Dr. Lewis. I don't know. That means class is over. Oops. We'll pick it up here on Wednesday.